Thank you. I left Los Alamos in 1975, well before Genbank. I moved here to NIH. It was actually Walter Goad who hired me at Lannell, so we go back quite a ways. The first I heard of Genbank was in late 79. I was at NCI. Al Rapson was my boss. I was interested in health science policy, and he detailed me to the office of the director, and I worked with Bill Raub. And it was Raub's meeting, it was a follow-up meeting to the meeting Josh Letterberg hosted that you heard about this morning at Rockefeller. Bill, who was then Associate Director for Extramural Research and Training, was actually trained as a mathematical physiologist. He had developed the profit system, which Howard Bolofsky was contract manager of. And the problem confronting the people at Rockefeller, which was the same problem that would later confront some of the people that are interested in the Genome Project, was how do you translate what they wanted to do into policy? And Bill was the perfect person to do that. So Bill hosted a meeting. Rich Roberts was there. Ruth Kirstein, of course, was there. And I don't recall, Elke Jordan was certainly was there. Walter Goad, Margaret Dayhoff. The one thing, I refer to Richard's remark about Ruth, the one thing I remember her saying after she made some introductory remarks is, I look forward to learning. Now, which to me indicated an open mind and that her ideas were probably evolving, and as we know, they did evolve in the right direction. And I think she's someone we should all be grateful to, and certainly also to her husband, Al, who was my boss, so I have to say that. At around the same time, he used to be my boss. At around the same time, Minoru Kanehisa wrote to me. Minoru was at Hopkins completing a postdoc. He had an interest, we shared an interest in DNA stability. He wrote to me about a postdoc. I couldn't hire him. I said, why don't you write to Walter Goad at Los Alamos? And they may have, in fact, I knew they had positions there. So Minoru went to Los Alamos, and 3 years later, he came back. I hired him at NIH. And Walter, when I spoke to Walter about how he was doing, Walter's remark was something like, Minoru set up the whole system. So Minoru Kanehisa was absolutely seminal in developing early versions of Genbank. He then came back, worked in my lab, and used the Framer system referred to this morning. Actually, he may have resisted at Los Alamos, but when he came here, he said, let's use the Framer system. I wanted to set up a database in the lab, and this slide shows something, this must be 1984, a paper we published in NAR. Was that before Richard became editor-in-chief? I don't remember. Rich, you probably know. 85. So this is just before Richard Roberts. And this is what we had set up in the Laboratory of Theoretical Biology, and we really had this version where we were taking protein sequence data, DNA sequence data, and tools, methods for analysis, which we were developing or importing from others. Now, in fact, the 79 meeting that Bill organized was a big disappointment to me. And this may bear on some of Bruno's remarks, because I refer to that as computer as filing cabinet meeting, because they totally ignored the possibility that the computer would be used for analysis. It was just thought of as a way to store data and retrieve it quickly and disseminate it. And I think what was missing there, and probably part of the reason people were reacting against this as just another kind of natural history exercise, is that they were just thinking about it in that term. This is a data repository. It was not just a data, it was, to me at least, and to some other people, and certainly people at Los Alamos, it provided the resources needed to begin to do deep analysis. And we all knew that computation, then at that time, a cray was a big deal, that a megaflop or something was a big deal, 2 megaflops. But it was clear what the trends were, and that we were going to be at a point where we could develop tools for really doing analysis. And I was doing some of that at the time, and when I'd speak about it, like how to identify exon-intron boundaries computationally, people would be very polite, and they sort of yawn. And so in 1984, any of us who were following what was going on in sequencing couldn't help thinking about where this was going, when we would be where. And The question is, and I remember Jake Mizell even, who then moved over to NCI, commenting, there's got to be a way to get there, meaning to have the whole human genome sequence. So I made this estimate and came out, and it actually agrees with something that Christian just showed, the second group of estimates he made. I estimated 2011. I look at this result and I say, this is a joke. There's no way, there's not a prayer that we're going to be there by 2011. And the reason I said that is because the reactions I got when I spoke about this, the culture just wasn't there. The biological culture wasn't there. Some discontinuity had to happen, and I hadn't the foggiest notion what that would be in 1984. So I just dismissed the whole thing, forgot about it. 1985, I got a call from Jay Keyworth, who was then President Reagan's science advisor, and asked me if I was interested in moving to DOE to direct their health and environmental research programs, which I did. And there I discovered that, so this is the summer of 85, fall of 85, I discovered, and I got this report from the Office of Technology Assessment, which spoke about sequencing the genome. And then I heard that, and I spoke to the chair of our advisory committee who told me about Bob Sinsheimer having a meeting. So then I began thinking, well, maybe this isn't such a crazy idea after all. And we had the first Santa Fe workshop, and that was March of 86, the same time Delbecco wrote his editorial. And these are just some of the people who attended. I guess, Christian, you were there. And Mark Patinsky organized it. He was the life science division leader. Walter was there. Walter Gilbert, both Walters, Gilbert and Goad. And it's kind of interesting, and I got a bunch of letters, reports afterwards, a few weeks after this, and it's kind of interesting to look through those letters. These are now in the archives at Cold Spring Harbor, and look to see what people were saying, because some of it was very prescient. So George talking about computation and something like 10 million a year, which happened to be just about where things were. Execution would require 10 to 15 years. Another one, again, these estimates of how long it would take to do something in the 10 to 20 year interval was a common guess of how long such a project. I think they were responding to my question of could it be done in the next 15 years. Then questions about how it would be organized, Cantor recognizing that the technology was going to change. I think Walter will end with Walter's, look at Walter Gilbert's remark, because it's interesting, because $3 billion was tossed around as quite a lot, and this is going to be the cost of the project. The fact is, Gilbert clearly recognized that technology was going to improve and change and the cost would come down. So $3 billion was an overestimate, and Walter thinks in terms of orders of magnitude. He probably, $1 billion was, if he were really pushed, I would suspect he would have probably said closer to $1 billion, which is just about where it was, and it was about where my guess was at the time. Anyway, we were all sort of guessing. I guessed in 1987, when I left the Department of Energy, that the project would be completed in around 2000, 2001. And the reason I guessed that guess was based upon an assumption that the economy would be more or less average. Nothing funny would happen. I was right for the wrong reason, because what happened is the timeline was, we got a budget submission. By the way, going from this workshop to getting something in the budget was not trivial. I mean, it was a big deal. The physics community was absolutely up in arms about the SSC. Remember, this was a time when the superconductor super collider was being pushed by the physics community, enormously expensive. They see this $3 billion figure, it's going to cut into the physics budget. So there was tremendous opposition at DOE, which not many people know, to this. The assistant secretary was committed to the SSC and never said no to this, but he also never said yes. I went to our budget examiner, and she was afraid to do anything. I asked her, could I speak to your boss? She said, okay. Our boss was Tom Palmieri. Amazingly, Palmieri bought off on this. I mean, I presented, and the reason is that I presented it for what it was. I didn't present it as a science project, because it wasn't a science project. It was a project to develop a resource. And Tom saw an end, or I presented it, and I said, this is going to have an end. Here's where it's going to begin, and here's where it's going to end. And so he was okay with it. He thought it made sense. Then Pete Domenici, of course, had national interest, but he was also a senator from New Mexico, Los Alamos, so Pete bought into this big time. And at that point, the assistant secretary didn't stand in the way and let it go, and we got a line item in President Reagan's 1987 budget submission of about $10 million, and that's what got this thing started. but it was, you know, I mean, people insulted me, they insulted my ancestry, and so on and so forth. The usual sort of thing that I think this is just a memo that I had written to the Assistant Secretary. So in 1988, so that, of course, increases data generation many fold now. It was expanding rapidly, exponentially, but now we're going to see a kind of a double exponential growth. NCBI was created. David Lippman was appointed director, and both events are seminal because I don't think anybody has this combination of grit and intellect that my friend David Lippman has. It's just remarkable what's happened. And Genbank really has made the Genome Project, is one of the key factors in making the Genome projects transformative. And of course, they feed back on one another. And it's also seminal because it has really made a change in computational biology. I mean, it's really introduced a discontinuity into computational biology. Now, mathematics and biology go back a long way, and I hate to say who was first, but I'll say Helmholtz, everybody knows. I'm sure there's probably some allusion in Genesis to mathematics and biology, so I won't say who's first. But Helmholtz certainly, in his study of energy balance in physiological systems, used mathematics and physiology. In this century, von Bekesi, cochlear mechanics, of course, won the Nobel Prize for that, and that involved partial differential equations. The Luria-Delbrück experiments and analysis is highly mathematical. Of course, Francis Crick's analysis. Who did he do that? Bruner, do you know who he wrote that paper with? Crick, and there were two other authors on that paper of diffraction off a double helix. So that obviously, Cochrane, right. Yes, you probably teach that for your course, right? Yeah. The tomographic problem by Cormack. I mean, there are all these singularities, major contributions of mathematics, but there were kind of singular events, and there was an underlying current. But this really was transformative in turning computational biology into a science. What was missing, well, before I go, what was missing, the question is, you know, where is it going to go? And the answer is, I have no idea where it's going to go. I mean, where's it going to go in the future? There are lots of futures, and I think maybe a lot of them will happen, and when they're going to happen, I have no idea. It's easy to think about things that can be addressed in the future, and thinking about them, it's good to inform one's thoughts by some of these remarks by Ray Kurzweil, who Kurzweil likes to think in terms of extrapolations, and some of them actually proved quite effective. So here it is. We achieve one human brain capability for $1,000 a year by 2023. And that's based upon, it probably makes that estimate based upon visual processing. That is, we'll be able to process at the same rate, the computer will be able to process moving images at the same rate as a brain can. We achieve one human brain capability for 1 cent by 2037. And you see, somewhere by 2050, we're talking about a human race capability on our belts and a laptop on our belts. Now, what the implications of that are, I don't know. But I have to say, I can think of a lot of problems right now where it isn't going to make a whole, problems that will make a difference, a lot of problems that won't make a difference because they're almost computational because they're exponential explosions. The thing about biology is not complexity, it's combinatorial complexity. So one of where things have moved ahead biologically in terms of making predictions and adding to knowledge and amplifying knowledge have been based upon conservation in one form or another. And that has to do with comparing sequences, or it has to do with what I'm going to talk about in a little while, finding binding sites on DNA, functional annotation, but it all involves conservation in some sense. Now, conservation is something, of course, that occurs not absolutely, but in a more absolute way in physics. And underlying that conservation are symmetry principles and group theory, which lead to powerful organizational principles. Whether, in fact, there are going to be similar powerful organizational principles in cell biology, I suspect there will be, and I think there's going to have to be, because I don't think things are going to scale simply with increased computational power. I think there's going to have to be very powerful organizational principles, possibly based upon conservation, possibly based on something else. And you see conservation in terms of of genetic circuitry as well. So it's there, but exactly what shape it's going to take, I have no idea. What was missing from all this, from these developments, was, and what bothered me even when I was here, Vince DeVita was director of the Cancer Institute, and DeVita thought, wouldn't it be nice to have a Cray computer and we could all do all these wonderful things. This is 1985, and Crays were the fastest machines at the time. And I thought, I'm not so sure, because you've got to have a cadre of creative users. You've got to have biologists who know how to use that machine if it's going to mean anything. And what concerned me, and I basically feel that most of the contributions are going to come from biologists. So what was missing, in fact, was a cadre of educated biologists. So back in the late 90s, Charles Cantor and I, Charles was then chair of biomedical engineering at BU, were having a conversation and thought we might develop a program in bioinformatics, a term I never liked, but it seemed to be fashionable. So we developed this program with support from NSF, and it's very much Boston, Cambridge-centered system in the sense that we have students at Harvard and MIT, and we share people on advisory committees and so on, but this is a university-wide program. It's been in place for 9 years now. Over 40 PhDs have graduated from the program, and over 70 master's degrees have graduated from the program. And it helps, and we need, and of course there are dozens of other programs that have sprung up in the meantime. So this is where education, I think, is going to become obviously become increasingly important, and it should be targeted at biologists. I must say that I had my eyes closed after I left Washington. Washington, I didn't think about genomics for 10 years. I went back. I was doing research in immunology and molecular immunology. And in 1998, when I started thinking about this program, 1997, I looked at the field, and it was just unbelievable. And I think one of the great stories in the history of science is the way the computer science community responded to this challenge. I mean, it's just absolutely been remarkable. And with tremendous sophistication. I mean tremendous, not just obviously computational, but tremendous biological sophistication, many of them. So it's a really remarkable story. And in the end, I think the Genome Project maybe have had more of a transformative effect on the culture of science than it will on the technical aspects of science. So that makes a big impact on the data. But let me say a little bit, so I'll say just a little bit about my own lab just to illustrate some ideas about conservation, a more subtle type of conservation, which is conservation of transcription factor binding sites. And I won't say anything, I'm not going to talk about methods. We've got two basically lines of research going on here. One is new engineering methods, and particularly in proteomics, developing label-free technology. And we first started doing this about 5 years ago. There weren't too many people doing it. Now there's a whole industry. In fact, there are several companies, including one that I'm involved in, doing this. And the other is computational work. And we're doing some work on new methods for annotation, a good deal of work on regulation, and still some work on infectious disease. And I won't talk. I'm just going to talk about, very briefly, I'll make a few remarks about kidney cancer and regulation. And so this will be work by Gul Kirka, who starred here, and Dustin Holloway. And interestingly enough, where's Dustin? Interestingly enough, neither of them are bioinformatics or bioinformatics PhD students. They were both cell and molecular biology students. And this is just the label-free technology, which I won't talk about. So first, I'll talk a little bit about wilms tumor. And this is just, what we were doing here is looking at human transcription factors for which at least a half a dozen targets were known. And the dark part of the bars show the number of targets that were known for a particular transcription factor. And the yellow part shows the amplification of that information, the number that were predicted at 95% confidence. And I'll just take one example, this WT1, the paper we have discussed at both Octima 4 and WT1. And basically, just to elaborate a little bit on Rich Roberts' remarks that there's a need for biochemistry because this is a tremendous hypothesis generator. So we've identified all these targets. You could begin looking at some of them, and you can immediately see provocative things. And the targets that were predicted, WT1 is involved in syndromatic Wilms tumor. And the targets that were predicted targets show up in the same pathways, very, very enriched in pathways that the known targets are in. So for syndromatic Wilms, WT1 is lost. Wilms tumor also, and that's a heritable form of Wilms tumor. There's also a form of Wilms tumor which is essentially the same clinically, but it's not heritable. It's sporadic. It's called sporadic Wilms. And for that, WT1 is not lost. In fact, it's normal in some cases, and in other cases, it's overexpressed. And WT1 is also involved in other tumors where it's overexpressed. So it's a murky situation, and it gets even more complicated simply because there are a lot of variants of WT1, a lot of splice variants. So this is what we predicted. This just illustrates amplification of information. This just summarizes some of what's known. So when it's lost or inactivated, there are a variety of syndromes, and that's invariably accompanied by syndromatic Wilms tumor. When it's normal or overexpressed, sporadic, which is the more common form of Wilms, and that's associated with loss of heterozygosity in a number of chromosomal locations and also loss of imprinting in some of them. And so how does all this stuff relate, overexpression, underexpression, normal? Well, if you look at the targets, it's kind of interesting. There are 9 targets in this region, 11p15.5, which is near where the Wilms tumor gene is. And there's LOH in those regions. If you look at those targets, some of them are oncogenes, some of them are involved in cell adhesion. They're all involved in physiological processes that are hallmarks of cancer. So it could be that this type of Wilms, the sporadic type of Wilms, it's nothing to do with WT1, with the transcription factor, but has to do with defects in the targets and targets that weren't previously identified. So these are things that need to be, that we plan to follow up on them, and I'm just touching the tip of the iceberg here in terms of what we've uncovered. And I think, there are people there are tens of thousands of hypotheses that things like this generate, and all kinds of people are making these hypotheses, and some of them more plausible than others. I could look at the top 1% in terms of reliability, in terms of confidence, and be even more confident. So, in fact, it seems to me we could use a resource. This is not quite what Richard had in mind, but it's similar, where these hypotheses are looked at and prioritized, and then they're tested experimentally, and then it needs to be funds for that. And then you'd really move a lot of things forward. And so a lot of it would be wrong, but a good fraction of it would be right, I suspect. I won't dwell on this. The idea here is that one of the targets is P53 of WT1, and when it's overexpressed, it could inhibit apoptosis by interacting with P53, and then there's a more subtle effect because another target is BAX, which interacts with BCL2, which inhibits apoptosis. So you have the same type of effect if WT1 is overexpressed. So inhibiting apoptosis, of course, apoptosis again is a hallmark of cancer. So it's what you might expect. Let me say a few words now about another type of kidney cancer, renal cell carcinoma, clear cell. This is, again, a molecular biology student, cell biology student, and I wanted to do something very simple. This last, by the way, set of predictions was made using SVM variants. There are 2 types of learning algorithms. There are supervised and non-supervised. So SVM was a supervised algorithm that we had validated, and I'm not going into all that. This analysis, I want to do something very, very simple because the data were very limited, and the student I had wasn't sure how. So I gave her the simplest thing I could think of, and I said, just look at all these samples of Wilms tumor, normal samples and tumor samples, and just look for genes that are always over or underexpressed across the whole population. So typically what you do is you look for genes that are on average overexpressed threefold or across the whole population, or have a p-value of 0.05 or whatever. And that's an average. So there may be, in that population, there may be cases where the gene is not over or underexpressed at all. But now this is a more stringent requirement. It's less stringent in one case where I just say, just look at those genes that are always overexpressed across the whole population. So you take a gene, and in every case, it's either always overexpressed or always underexpressed in the tumor relative to the normal. So she did that, and then we looked at the distribution and identified those that were at the 99th percentile in terms of the difference between, so you'd have, if something is over, let me see, go back to this last slide again. So this illustrates it. So this is, for example, a typical gene, and you see in this case, the gene is always lower in the tumor. In every tumor cell, that's these Ts, in every tumor cell, the gene expression is lower than in every normal cell. And now you look at the minimum difference here. So this is the maximum it is in any tumor, and this is the minimum it is in any normal. And then you look at the distribution of those distances for all genes, and you do some randomization. And then you pick genes that are out at the 99th percentile. So those are the things, those are the potential markers. So this is a really simple exercise. There's no fancy mathematics in this at all, except we did something similar with an SVM, and the results are pretty concordant. 18 out of 20, you get the same result. So now the question is, so we have these potential markers, and the question is, how do they stratify things? And you could do this in a couple of ways. So here's hierarchical clustering. The dark means normal, the green means downregulated, the red means upregulated, and across the top here, you see the samples. N means normal, and T means tumor. So you go from normal as you go across here to tumor, and you go from low-grade tumor here to high-grade tumor there, and there's one outlier a little bit right there. This is a low grade, and it's mixed in with the high grades. But other than that, the markers stratify. Now, if you look at the principal component analysis, see the first principal component separates tumor here from normal here very, very cleanly. The second principal component right here separates the high grade from the low grade, except for this outlier, except for this one outlier here, which is a low grade mixed in with a high grade. And the third principal component here separates these two normals from this group of normals. Now, these normals actually come from, and this is something we need to follow up with the pathologist on, these normals come from people who have high-grade tumors. So they may not be normal. That may be what's happening. And if you look at where they lie very close to on the other plot, they lie very close to the tumor. So this may lead to a reevaluation of the data. This just shows, very briefly, that the 158 genes occur at a they have cancer genes. Compared to OMIM, they're highly enriched with cancer genes. And we don't need to spend much time on that. What this shows is a clustering by process. So there are, I mean, Weinberg has written a very nice paper on this, and there are half a dozen key physiological processes here. And you see one is, of course, growth self-sufficiency. And you see this cluster up here. Red means upregulated. Green means downregulated. Small circles mean predictions. Large circles mean known already. You've got this hypoxia-inducing factor. Of course, this is important in angiogenesis, which is another hallmark. Adhesion. There's SFRP. This is escape from control. So soluble fissile-related protein. This is a Wnt pathway inhibitor. So when this is down, the Wnt pathway becomes active, and that, of course, it's a developmental pathway, and of course, it's implicated in cancer as well. So there are a number of provocative predictions here. So what we wanted to do was see if we could understand, and there are a lot of mechanisms for downregulation. We decided to look at one of them, one possibility, which is overmethylation of the promoter. And we did that using MALDI-TOF, and found, we took 20 genes, and we found 7 of them were hypermethylated promoters. So 20 of the genes that were downregulated. And this is a summary of them. And then we looked, then we did Western blots on these, and that last column shows the significance level. So there are 3 genes. SFRP is certainly the most dramatic, both in protein level and in gene level. But then there are a couple of, and SFRP makes a lot of biological sense. Then there's another one, SCNN1B, and I'm not sure how much biological sense that makes. Cation transport, it may or may not, and that's clearly significant. And synaptomatogen 6 is significant, and the others are marginal. So there are at least 3 potential markers. And for this to be of any use, now there's a very easy assay. If you did the thing about renal cell carcinoma is you can screen the urine. So you can look for markers in the urine. But if this is going to be a prognostic, what you need to do is test this on a population like the Framingham population, which is what we're doing, which has had about 90 people show up over the years, over 3 generations with RCC. And you look at them when they're showing up normal, and they're normal, and now they have, some year, they have renal cell carcinoma. And the question is, if you look back in the years that they were normal, do you find that this, and you look in the blood, do you find that, or the urine, do you find that this is downregulated? So if you do, then it really becomes a useful tool. But we're not there yet. But that's basically the strategy. Okay, just to summarize, here, thousands of previously unknown targets for 152 human regulators were identified at 95% confidence. That's right, that's correct, yes. Starting with 15WT1 targets, 354 targets were identified at 95%, 111 at 99%. So there's a lot of stuff to look at there, and I just looked at WT1, but I could have looked at any of the other 100, and we could have told stories about those. Chromosomal loci, 11p15, 4p16, I didn't say much about this, 1p36, were all previously shown to be associated with LOH and LOI and sporadic wilms, and they're significantly enriched in WT1 targets. The targets are localized to cytobands 11p15.5, 4p, well, you can read this as well as I can. I didn't go into detail on any of it, and I'm not going to even bother reading this, but the basic idea here is conservation, and here it's a weak conservation because it's binding site, which in fact, it may not even be sequence, it may be stability, which is one of the things you train on. So if it's stability, if it's free energy, for example, well, there's a lot of degeneracy between sequence and free energy. So sequence by itself wouldn't work. So that's why you'd have to do more than just sequence alignments. That's about all I want to say. Thank you very much. It's been an exhilarating experience watching Genbank develop and watching it develop here with David and certainly serving on his committee, and Don Lindbergh's support has been instrumental. And of course, the staff, the senior staff, the leadership, without that type of staff, nothing would happen. Thank you all very much.